look at problem number 1 here. It says DNA samples taken from rats. This is a released question, by the way. This would be a computer question, so you got to expect. You can go online at the OST, and I'll put the link in this review when I post it on Schoology. But you can go online and actually do this, like choose which one it goes with which one. They'll literally ask you to pick it up and put it in the appropriate space. The DNA samples taken from rats from the same colony produce 4 gel electrophoresis bands. Remember we talked about in the earlier explanation and in your class reading, gel electrophoresis is when you can take proteins or DNA and you put them in a well and then you run the well and you get a pattern. The pattern corresponds with similar proteins or similar DNA molecules. Similarity reflects a similarity in the DNA sequence, in the RNA sequence, or in the protein amino acid sequence. So depending on what you're running in the electrophoresis. So DNA samples taken from rats from the same colony produce 4 gel electrophoresis banding patterns shown. So you got RAM B, C, and D, and there's A. Determine the relationship between rat A and all the other 3 rats, B, C, and D. So one of these is an identical sibling, another one's a sibling or a parent, and the third is completely unrelated. Now, to be honest, even though the details are complicated, the reality is pretty simple. Which one of these 3 matches most closely? Which one of these 3 relationships is the closest relationship? So the closest relationship is going to have the most closely related pattern, and the one that's unrelated is going to have the furthest pattern away, and the one that's in the middle will be in the middle. So place the relationship in the labels of blank boxes under the electrophoresis patterns for rats B, C, and D. Place only one label in each blank box. You may use the label more than once. So where would you use, let's take a look at this. This particular rat A has a big pattern, 2 little ones, then 2 same widths, and a third one that's thinner in about this area. You'll see A and C have identical patterns. Identical. Well, I'd have to go with those 2 being identical. So identical would go here, right? I would think. Sibling. So a sibling is identical would be to rat A. The next, so that takes care of identical, that's moved, and I can use it, you place only one symbol in each box, but you could use the label more than once. Okay. Well, one would assume if they had 2 of them, they were all 3, this C and D had the same identical, they'd be triplets, right? In this case, they're just twins. Well, I look at A and I see that this is the same, and here there's only 1 band instead of 2, and that band is down here in this one rather than being up there. Then you have this band is the same, but then it's missing this thinner band, and there's a band that's a little thicker that's down lower, and then there's a third band that's thin here but thick here. So this to me seems like it's very similar. They're similar but not quite. How similar are they? Well, let's count them. This is 1 band that's the same. This is the same. I'm just going to mark the things that they're similar or the same. Here's one. One of the 2 there is similar to that one. And then this band here is still in the same place here. And then this band here is similar. It's in a similar place. It's thicker on the right than it is on the left. But to me, there's 1, 2, 3, 4 similarities between A and D. So there's 4 similarities. So at that point, I'm going to have to say it's similar enough that it could be a brother or parent. Sib or parent. And the esports team is practicing in the room, so you're going to hear talking. You can't ask them to be quiet. I mean, they are. It is a sporting event. All right, so that takes care of 2 of the 3. The next question is this rat B, is it a sibling or parent? We know it's not identical. Is it a sibling or parent, or is it unrelated? Well, how do I know it's not identical? Well, look at it. It's not identical to this. Now, this one is similar, but it's thinner. It's in a similar place, but it's thinner. Here, there's two thin ones. Here's a real thick one, and then this one's lower. It's not even in the same spot. It starts earlier, ends about in the middle of this. This one begins here, ends down here. Very different. Then this band here is much higher than this band is. This one is about at the same place. It's thinner, and this one's thicker. And this one is just as thin, but it's even in a different place. Is there any similarity between B and C? I don't see any similarity between B and A and B. There are four similarities between D and A. You can argue some of those, but there's at least a couple similarities between A and D to four similarities between A and D. A and C are identical, but A and B have no similarities as far as I can tell. There's not a single band that's the same. So I'm going to say no. I'm going to have to choose unrelated. And to get 1 point on this question, you have to pick up identical sibling, and it has to be put here. Sibling or parent has to be put here, and unrelated would have to be put there. You'd have to put all three in the right place to get 1 point for this question. So that's how tricky these can be. All right. And the answer's down here for the scoring guide. But that's the reasoning. That's how it works. Okay, so we're looking at this. We're looking at amino acid sequence differences, and they're going to look at differences, and that's key. And we're going to try to figure out which ones belong in each of these boxes. These two are the ones that have the least amount of difference, right? Which two on this chart have the least amount of difference? Well, B and C have only one difference between them. So these two have to go here. So B, if I put B here and C here, that's reasonable. They're shared only one difference. See? So B and C, now I don't have to worry about B or C. I just got to worry about D's already placed, right? So D's there already. And the question is, does A go here? Or does A go here? Where's that going to go? Well, how closely related is A to D? A and D have 11 differences. A and D have 11 differences. How closely related is let's see, which one's left? How closely related is B, C, A, B, C? Well, how closely related is E, which is the other difference, E to D? E and D share 3, so they're really closely related. Where A and D have 11 differences. So what if we placed E here? That means there's 3 differences between D and E, so that would be 3 differences. Just like there's only 1 difference between these two. But A has, that would mean A would have to go in the only slot a bit left to it, which would be here at the bottom. And you have to ask yourself, is that right? Does that show a lot of difference? How closely related is A to any of these? Well, A is 11 at the closest and 14 at the furthest away. So that makes sense that A is all the way out here. D and E share 3 differences. There are 3 differences between them, so they're very closely related. But then you look at D and E, and between D and B, let's say, between B and D, there's only 9 differences. That's a lot smaller than 14, like between A and E. So all this makes sense. This is the best arrangement that you can make using this one chart. The key here is that you're just looking to see, why is this tricky? Because normally we look at how many different common things they have when we did a cladogram. Here they threw you for a loop and they said how many of these are different. These are the differences. So the larger number is not an increase in similarity, it's actually an increase in differences. So they're going to be the ones that are furthest away. So A being different from B, C, D, and E, the most different from all of them, should be out here. B and C are very close to one another. We can see there's only one difference. They should be together. And D and E should be together. Notice they have a common ancestor right there. They only have three differences. So this setup here shows clearly that B and C have a common ancestor recently. D and E has a common ancestor relatively recently. Where A and the rest of them have a common ancestor way, way back in the evolutionary history. When we look at this, the scientists have found an allele of a plant gene that may protect plants from some types of insects. To determine whether this allele is present in other plants of the species or is in a recent mutation, they conducted the following experiment. They isolated DNA known to contain the allele from one plant that is unaffected by insects, and that DNA-resistant sample, I think it should be obvious, is listed here since they actually tell you DNA-resistant sample, right? So this is your DNA-resistant sample. Then it says, isolate the DNA that may contain the allele in three plants of the same species growing in the same location. So there's 1, 2, and 3, right? In the same location, so you're kind of controlling all the different variables. Treat the resistant DNA sample with the 3 DNA sample and the 3 DNA samples of other plants with the same restriction enzyme and generate smaller segments. So you're breaking this enzyme, you're breaking the DNA up into small bits, and you're running it through a gel and separating. Again, what are we looking for? Just a pattern, a similar pattern. Which one of these has the most similar pattern or different pattern, depending on the question they're asking us? So they're just taking DNA. Remember, DNA is very long. And restriction endonucleases or restriction enzymes are taken from bacteria, and we can use those to cut DNA up into different size pieces depending on the sequence. So wherever you see T, T, A, C, this one restriction enzyme will cut that here, where another restriction enzyme might be looking for C, C, T, A, and it would make a cut. It would make a cut here, every CCTA. So the sequence, depending on where they cut, it tells you something about how related the two samples are. And then you run them on a gel, and the bigger the piece that's been cut, the further up here it is. The smaller the piece, the further down here it is. The bottom line is that the similar patterns show you how closely related that individual is to the reference, in this case, the DNA-resistant DNA sample. All right, so they treat the DNA-resistant sample and other samples of plant restriction, although they run a gel electrophoresis DNA segments, analyze the result of gel electrophoresis. Click on the sample that has the same allele as the resistant plant. Which one of these has the same allele, the same DNA sequence based on this data? They didn't sequence it; they just broke it up in a regular pattern. And they asked which one has the same size patterns. Well, is this the same as this? I say no. I think it's clearly no. And I think you can easily see that even though this one's common in most of them, I think it's easy to see that this one's also different from this one. So this is also different. The only question is DNA sample 2. Does DNA sample 2 have it? And if you look, this is the same, 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 same. So this has actually the same allele. And Bob's your uncle, there it is. This is the answer. It's 2, DNA sample 2. Why? Because when I took an enzyme, they cut in between TTAC, in between the A and the C. And sample 1 had a T T A C T T T T A C C C A T T T C. Well, this is just an example here. Sample 1, every time there's TTAC, was cut here, was cut TTAC, was cut here, TTAC was cut here, TT, and I would say the corner of this. So if I cut this sample with an enzyme and a restriction of nuclease, an enzyme that cuts the DNA in these particular sequence, then I'd have three sizes. I'd have a banding pattern that looked like this. One big one, that's this one. This is the big one. There'd be one medium-sized one here. That's this one here. And then one little one that would be all the way down here. Again, the smaller it is, the further down the gel it goes. On the other hand, if that same gene for a different species had a T T G, right? T T T A C C A T T T C, right? Then you would find that there would be a TTAC here, and they'd cut it. It would cut there, which would only give you two bands. One band that would be this size, and one band that would be this size. So it would be very close together. There'd be one band that would be one band that's smaller, slightly smaller, so it would be a little higher. And there'd be another band that's just very close, but still below. That would be the pattern for this particular example. And then what if you had a third example, like the problem we just had? And this third allele, had A, T, T, C, let's say T, 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 A, G, C, A, T, T, C. Well, that would have just one band. It would just be one band because there is no TTAC. Nowhere in here do you see TTAC. So there would be that would only have one single band in its gel. So if you ran the three gels together, which you'd get if you ran electrophoresis, you'd get one column that would show this banding pattern, one column that would show this banding pattern, that was about right, and then one column that would show this banding pattern, really big piece because it's not broken up at all. So you'd have in a gel electrophoresis that had something that looked like this. These are the banding patterns you'd expect if we did it this way, but they broke it up, and this is what they got. They used a restriction of nuclease. They cut a bunch of different places, but everywhere it cut, it's only going to cut between two specific letters in the sequence. Everywhere it cut, wherever it cut this one, you're comparing to these three samples. The most similar one is this one, so these two have the same alleles. These two have something different about them. They don't have as many, or they have more, or what have you. All right, let's go on. Okay, so let's take it. A mutation occurs in the DNA base sequence. So you get GGG, GAG, TTA, resulting in the base sequence this. So what's the mutation? Let's take a look at it. Where did it mutate? It's GGG. So this last G went to an A, right? That's a mutation. This A is different. GAG is the same. TTA is the same. So we changed this piece of the DNA. Now, what if you would have turned that into RNA? And let's forget about cutting it out. What would happen if you turned that into RNA? Well, RNA, this RNA would be CCU, right? Then the next three in the RNA, assuming it's not an exon or an intron. Let's assume that everything is in the exon, so it's going to be in the mRNA. Then we have the next three is going to be CUC, and the last one's going to be AAU. All right. Use this information. So we what did we do? We changed it to an RNA, DNA to RNA. We transcribed it. Now we're trying to translate it. So it's asking use the information table to determine the effect of this mutation on the amino acid produced. They give you the table and tell you UUU is going to give you phenylalanine. There is no UUU here, so we don't have to worry about that. CCU is going to be proline. CUC is going to be leucine. AAU is going to be asparagine. By the way, we'd have the same. This is the mutated version, right? Mutated. The non-mutated would be CCC, CUC, AAU. So if we translate this, this would be leucine. We put it the amino acid sequence in different color. This would be leucine. CUC would be CCU is proline. I'm sorry. I'm trying to do three things at once here. Proline, and CUC is leucine, and AAU is asparagine. Now, CCC, on the other hand, in the non-mutated version, is proline. I'm sorry, CCC is not proline. Oh, yeah, so CCC is proline. CUC is leucine, and AAU is asparagine. So you have exactly the same amino acid sequence. Do you see that? You have exactly the same amino acid sequence. There's no difference. This is the same as this. There is a mutation in the DNA. The mutation went from a G to an A. That did give you a different RNA, but because of the redundancy of the genetic code, it did not change the amino acid because there's two ways to get proline. Do you see that? There's two ways to get proline. So because there's two ways to get proline, means that you can have the mutation, but it not change the amino acid sequence. We call that a silent mutation. It is no longer, even though there's a mutation, we don't see it. So it's called silent. So what's the effect of this mutation on the amino acid produced? Proline is now lysine. That's not true. We just said that proline did not change. CCU is the same as CCC. So that's not true. Proline is now lysine, and asparagine is unchanged. Not true. Proline is unchanged, and asparagine is unchanged. There's our right answer. Proline is unchanged, and leucine is now phenylalanine. Well, that is again incorrect. So just by translating it and reading it, you didn't have to know it was a silent mutation. I'm telling you that, but you didn't have to know at that point. But it did not change the amino acid sequence, so you keep the amino acid sequence the same, even though you did mutate the DNA. All right.